Okay.
Look forward to Basel.
Thank you all very much for joining. If we could have your... Thank you for being here. My name is Frank Locke, and I'll be the moderator or player coach in this session. Precision health, AI, how can it contribute to advancing medicine and obviously ultimately to patient care? Interestingly, we just had an extended round table with about a dozen experts in this field. We had some very in-depth discussions, and my colleagues here on this panel will try to summarize that a little bit. I have a couple of questions for the panelists, and then we look forward to your participation and to your questions. So let me give you a bit of an overview of what we just discussed, at least my 2 cents, so to speak. This was very much about integrating. Well, my perspective is we come from this quarter century of the genomic medicine. 25 years ago, the world was very optimistic. We would have the blueprint of life, the book of life, because we had the human genome. That those expectations were too high. It has delivered, but it has not nearly delivered as much as we were hoping. It turns out we need a lot more than genomic information, and we're not only talking about epigenetics and and transcriptomics. We're talking about proteins, eventually protein forms, multi-omics. We need multimodal information. We need interaction. We need structural data. We need spatial information. We need longitudinal time course information. So the complexities of the post-genomic era, if that's the next 25 years of understanding disease biology and doing better drug discovery, it's absolutely daunting. The complexity of information that we have, and by the way, the big gaps of information that we don't have yet, where we don't have good lampposts to get all the information that we would need, provides enormous challenges, provides enormous opportunities. It's a sobering reassessment of how much we know about biology and how much we don't know, and why our drug discovery success is still limited. Anyway, but it is the confluence with AI and deep learning. I don't think we could do any of what we're doing without AI and deep learning. AI and deep learning also isn't enough. We need good data. We need more complete. We need multimodal phenome information. And we're very focused on humanized data, much more so than mouse models or animal models in the past. That's my quick preview. And with that, I'd like to ask my 4 colleagues who were all on this panel earlier this morning to give us their 3-minute summaries and perspectives. And I'm old-fashioned, so I'll go with ladies first. Carolyn, would you start?
Very good, thank you. So what we discussed this morning, you related to it already, is we have a lot of data, yet we have very uneven data. We discussed that the early drug discovery space is really rich in multimodal data, and molecular data, like the clinical space, is starved for it. We have a lot of textual data, and we use AI on both sides for a long time. On one side, to gain a better understanding of the disease. In the clinical side, to increase the efficiency of process and reduce cost and go quicker. And there is this discrepancy now and this ambition to bring the molecular data and the disease knowledge to the clinic, not to go quicker and cheaper, but to increase the probability of success. So that was something we discussed. We also discussed that there are really pockets of excellence and building blocks in place to create virtual cells and be able to inform the clinic, but that for the moment, because this field is just starting, even so it advances quickly, they are very compartmentalized. So we discussed on how to bring the people together across academia, across hospital, across pharma and biotech and biobanks. And we discussed a lot about the AI evolution, so I can pass to Fabian.
Fabian, if you want to continue.
May jump in.
Yes.
So I like that sort of the original vision to some extent could maybe be, can we understand patient variation, right? I mean, let's say if any one of us goes to the hospital, either you don't want to be at the mercy of the current treating doctor and you remembering what type of sort of trajectory you had before to be actually diagnosed. What we actually want is some type of like a Google Maps sort of thing that tells us not just where we are with limited amount of data, where we can sort of then fill up all the other sort of missing modalities. In our case, we discussed about omics and so on, but it could be also all kinds of things that sort of goes there. So we don't want to just learn where we are, but also where we go. I like to call this patient landscape, sort of finding this type of state, but then also the type of trajectories that we roll down. In the sort of equivalent for cells, we often call this Waddington's landscape. You have heard about this. So let's say we have the Zugspitze. This is where sort of the fertilized egg cell starts, sort of rolls down the various valleys, and then sort of makes those different cell types that sort of makes up our body. I think similar to that, it would be nice to sort of have maybe some trajectory where it's sort of healthy, when then sort of we branch off into various disease states. And maybe we can also sort of understand those branching processes. Maybe we learn where those branching points are, zoom in, and then see what type of maybe mechanistic perturbation. So it's not just the map where we are, where the lanes go, but also how we sort of switch lanes. That could be a treatment, could be something prevention, but I think sort of putting this together. So how do we realize this with AI? For me, such a patient landscape, which is essentially a patient representation, it's a sort of classical machine learning type of problem, that could be the highest level in a multimodal, multi-scale foundation model. So this could be sort of the top thing of what we like to call internally a virtual human model, where sort of the top layer is a readout on the patient level. But then let's say you have something on the imaging or sort of organ side, something on the tissue side, something on the cellular, maybe even molecular side. What we have at the moment is that we have very good individual deep learning models. Those are often called foundation models, right? That sort of tie those layers together. But can we now bring these together? How can we bridge the scales? There's some first very strong evidences that these things happen on the sort of smaller scales. We and many others build virtual cells, and here's a true expert, Steve Quake, who has been pulling a lot of us in the community together to really build these. But can we then scale them up across it? And I think it's a fantastic sort of data-hungry type of endeavor. But if we can get that, this readout, this link to the phenotype, which is, I think, one of the holy grails in our field, right, could potentially be achieved with that.
Eike, do you want to continue before Steve disrupts us all?
Sure. Yeah. Yeah, let's bring us back to reality here. So if you go to the hospital, when do you go to the hospital? It's not when you're healthy, you're going to the hospital when you're sick, right? So that's a fundamental problem that we have. You wait until symptoms appear, and they say, oh, something is wrong, I go to the hospital. And then you have physicians, they look at you and say, oh yeah, you have this aches, and maybe some markers is up. But diseases are mainly phenotypically defined. So how do you make a model of phenotypes? You could do. It's not very helpful, I think. So we need to break those walls, basically, in medicine, because right now, medicine is not set up for what you want to do. It's very nice that you want to do this, and I think it's the right way to do it. But it starts with the blood draw. If you take blood, the nurse takes blood, so what happens? It's sitting around, and then they collect a few things, and then they ship it to a lab that's maybe somewhere else, maybe in a different city, maybe 30 minutes away. So during that time, immune cells, which are important for the studies we want to do here, they will die, And then the other ones that are not dead yet, they respond to the dying cells. So there's nothing you can measure in terms of multi-omics, what we need for this type of data, if you do it with like standard way of practice at the moment. So that needs to be fixed, meaning this pre-analytics, you have processes from the blood draw to the analysis has to be short, has to be done always the same way, so you actually have reproducible data that you can then learn or use to learn this type of machine learning techniques. So that's one disruption we need in hospitals and physicians. And so that's important, though, because in the end, the other disruption we need is that the data that we all have, like we know our lifestyle, we know, our watches, not your watch, but your watch, they measure things all the time, but who takes those things into account? You yourself, there's a little app for it, but the physician would need to know, right? They don't have this type of data, and they don't know where you live. Well, they know, they can look it up. And if you look at the data from large trials, what makes you sick is the postal code. So where you live means how you behave most of the time, what the environment looks like. So all this type of data is not taken into account in hospitals because it's too late, and you always lived there. That makes you sick, and that was your lifestyle, that was your environment. So I think to really make truly preventive medicine or preceptive medicine, like we call it, we have to intervene much earlier. I'll give you an example. If you smoke, your likelihood to develop obviously cancer is higher, but also rheumatoid arthritis. It's the most important risk factor for rheumatoid arthritis. Why? Well, if you smoke, that means you have something in your lungs and the immune cells respond to that, and you make autoantibodies. And that 1% of you have this. 1% of the population has these autoantibodies, but you're not sick. You don't have rheumatoid arthritis. But over time, you get more and more autoantibodies, and then you develop the symptoms. We can now see already, 2 years before someone has disease, that some person will become sick and others don't. So we have predictive measures using omics to say this person is going to be sick in 2 years. Let's treat him before we actually is getting sick. So we do this with atherosclerosis, right? High cholesterol levels will make heart attacks or strokes. A third of the population will die from it. Look at your neighbor, one of them. And there's drugs you can take. You can take statins. There's now PCSK9 inhibitors. There's things that lower the amount of cholesterol because our environment is helping us to get sick. So I think we need to really rethink medicine, and then once these things are done in the right way, you create enough data that are good enough to train these models.
So we have an addition to the panel. Delighted to have Steve Quake from Stanford University here. I believe for some years you were also in the Chan Zuckerberg Initiative in a leading capacity, and you contributed this morning. Please contribute now with your perspectives.
Well,
Steve.
Thank you so much for the spontaneous invitation. I guess I'd start off by chatting a bit about one of the things you said this morning, which I thought was, really right on, and a shared belief that this metaphor of the human genome as the blueprint for the organism is completely incorrect. The proper metaphor is maybe it's a parts list, but we really...
A dictionary.
Yeah, exactly.
Yeah.
And, we really, despite 2 decades of having the genome in front of us, are very limited in our understanding of how to use it. And it's been powerful, but there's so much more to do. And the example I always talk about, and what I'll be actually the subject of my talk on Sunday, is even something as simple as trying to predict the cell types of the organism from the genome, we can't do it. It seems like in principle that ought to be a solvable problem, but we're not there yet. And the last decade of my life in the Chan Zuckerberg world was around trying to solve that experimentally. I mean, that was the motivation for trying to measure the transcriptomic cell atlases, to do experimental characterization of a molecular definition of all the cell types, because we couldn't predict it. And it's possible that, those data sets will enable us to build AI tools to do that, but we're at the beginning of that journey. And I think it's also important to recognize, we talked about this morning, that the genetics are only one component to disease, and trying to measure phenotype, or the things that have happened along the way, the things we ate, the things we were exposed to, none of that comes out of the genome. Like, I sequenced my genome 15, 20 years ago, and it says nothing about the effect of all the chocolate I love to consume and what that means to my health. And so these other, what you call multimodal ways of measuring, are very important in understanding our health states. It's not enough to look at genome or transcriptome, but to look at the proteins, the metabolites, the changes that have happened. And those sort of data sets together are going to be very powerful in sort of the basic science discovery of causes of disease and how to treat them. And AI tools are going to allow us to sort of integrate that together in ways we never have before. And that's like a revolutionary thing for science. The final thing I'll say is that part of it is sort of the discovery part. The healthcare delivery part of it, we talked a bit about this morning, is a different sort of challenge, in that how do you bring the fruits of those discoveries to the 8 billion people in the world who need their health treated? That's a different problem. There the problem is, what's the least you can measure about a person in order to determine how to treat them because of the cost of measurement? That's also a problem that AI is going to be revolutionary for. It's a different sort of inference problem, but one that's equally important.
Yeah. No, I would add to that even, there's so much more biological information than what's in the genome or even the epigenome or the transcriptome, there's much, much more information. They're at different time scales, and it's a feedback loop. The central dogma of molecular biology was great because it clarified the primary direction of biological information flow, but it also has been, and when something's called a dogma, you should be careful, right? It has also done us a disservice because it obscured conceptually for many of us for a long time how much interaction there is and how much environment can interact with the phenome, and the phenome, we have reverse transcription and eventually integrase and whatnot, can actually modulate what's going on even at the genomic level. So, welcome to this beautiful new world. It's daunting, it's amazing, but it gives us so much more opportunity. So where are we in this journey from genomics to epigenetics, transcriptomics, the other omics is the spatial interaction, structural biology? Where are we in this? Are we 80% there? Have we gone from 10 to 20%? What are the big gaps? Would you like to, maybe in reverse order, we'll start with you, Steve.
I mean, just to jump off again from the central dogma, I mean, Crick's paper there, I went back and read it recently. He was concerned about information flow in a very narrow sense, just about information flow to protein synthesis.
Right.
We now know there's many other important aspects of information flow in the cell and in cell biology. And so it's a much more, as you were saying, a much more general problem we're facing now, and I think have the ability to take on and make progress with. And so I think we're in a revolutionary time in cell biology. It's sort of an amazing time. The discoveries are going to be just fantastic over the next decade, and many of them will center around these questions of information flow in the most general sense.
Yeah.
So I feel like we're not, I don't know if I could say percentage of the way there. The next decade are going to be amazing. Let's put it that way. I'll leave it with that.
Caroline, you want to give us your perspective? Where are we in this journey? How much do we know? How much do we not know?
Exactly. We know better what we don't know, to take somebody's saying this morning. We have loads to go. I think technically AI is evolving fast. We can do a lot of very cool things. The question is on what data do we do it? And it's true, we have a lot of molecular data in the early space. Most of these data come from cell line experiment where we have profiled the heck out of them. Coming back to the pre-analysis or pre-processing and how the samples are important, there was recently a presentation from Rebecca Gundry from the US. She's getting hold of human heart twice a week, and then she took 10 years to optimize a protocol where she can profile what's at the surface of the cell in there. And one of her main findings was you put those cells in culture one time, you passage them, you change by 50% what's expressed on the surface. So we need to keep in mind that we don't only need data, we need very good data to go forward.
Fabian, you're on a roll.
I think the dynamics part is fantastic, right? And it's really so lovely to add. When we started to set out in the Human Cell Atlas, the sort of the spiritual successor, if you want, of the Human Genome Project, right? It was essentially enumerate all the variation in the human body. And, even there, we came up with some type of metrics to see when are we done? Because that's kind of what you ask, right? Like, how far are we? It turns out it's freaking difficult to actually be good about it. Like if you look at the first human genome even, right? I mean, what was the N number? It was like 3 or something like that, right? And still nowadays we're doing versions of GWAS and adding more genetics and sort of getting variation in. So I think there's always like another level of variation that sort of makes your complex, the problem sort of exponentially more complex. But I think so one thing, for example, we can, I think quite safely say is, let's say if we look at the blood or something like that, right? We have a lot of, let's say, single cell description of blood variation, including RNA, maybe some proteins. Of course, it could always be more, right? But I think in a control, maybe there sort of some type of saturation might be achieved with some type of modalities. But there's sort of new angles coming in. Within UK Biobank, for example, we're contributing to a project where now for 5,000 of those subjects, where you have all this deep genetic characterization, this phenotypical characterization, get also single-cell RNA-seq in the PBMCs. So even though we have so many PBMCs, you can sort of add that variation on top, and you can start these additional analysis. I think what's important to say is, I think experimental space is exponentially hard. So even stuff that you can measure, but let alone what you can do to things, is something we can never even be roughly there. So we had just one paper where we tried to then fill up that space computationally. So we measured like 0.0001%, but then we went to all of Campbell and sort of added that by imputation. So I think we can never measure all of us at all time points with everything. Even that is just going to be a little bit. But I think the rest, it's quite safe to say that we can, with some sort of error, know maybe roughly where we are. And I think this might be more than we have. It definitely works very well for cell lines. So if you have a bunch of cell lines, you can, to some extent, impute to some others. We can maybe take this step a bit to organoids. And more recently, we had a paper together with a colleague from ETH, where we were able to sort of map them, in this case, brain organoids to also human development. So there are some links where you can also then start enriching also these more complex situations that, human developmental brains very hard to observe in reality. So do the perturbations on the organoids, transport them up with some error. I think it's getting there, but of course, small percentage.
Yeah, so you ask where are we? So I think we are not that far in terms of where we want to be, but still, in science, there's a lot of new technology, a lot of knowledge, and with AI, there's possibilities to really integrate data as we couldn't have done it before. But I just want to explain the gap between the science part and the vision that we have, again, with medicine. I used to be intensive care physician, so people are really sick. They have an immune problem, right? They're really sick. What do we measure? High CRP levels. How many blood cells you have? Oh, you have too many monocytes. It must be an infection. Not necessarily. Neutrophils, too little neutrophils, not good. So very rudimentary things are measured these days in the clinic, not just because only the pre-analytics, but we don't translate these things to the humans that are really sick in the clinic. And that's actually why I left medicine, because in the end, we just kept them alive as good as we could. Some fluids in, something for the heart, some pressors by drip. Then you pump air into the lung, put some oxygen in, so keep them alive. But nothing was done for the real problem that these patients had, which is an immune problem, right? And now we're at the stage where we really understand how immunity functions mostly. The cells, took like 100 years to find all the different cells. We know how they react. We know the components, why these cells react to triggers. We know their language. They make cytokines. They kind of communicate. If you interfere with the language, you have massive effects on many diseases. But there's a big gap between pharma development, science, and then medical implication. So if we only take a fraction of what we can do in science and apply it to the medical field, we would be completely different. We would have molecular information on diseases, not phenotypic. We would then say, oh, this molecule could be working in that disease. So we're a little bit in this direction in the cancer field, where you can now just sequence, and you have all these mutations, and say, okay, this is the kinase here, kinase there. Maybe this triple combination would be good for the patient. So we're kind of ahead in the cancer field, but in inflammation, and 75% of diseases are driven by inflammation. I'm an immunologist. So
75%?
75%, even many cancers. We're not there at all. We measure CRP and how many blood cells there are, like Rudolph Hirsch, you have done it.
Yeah.
So that's a big problem.
So I'll do one more question from me as moderator, and then we'll turn things over to you. So get your questions ready. We've been talking a lot about humanized systems, right? How I deal a lot with oncology research and cancer biology, and in the cancer field, there's this by now old joke, oh, the good news will cure all cancers in the next 10 years in mice. And then 95% of the translation into humans fails. Of course, in neurodegeneration and many other areas, that's worse, perhaps. At least I guess our brain is very different from that of mice. That's somewhat reassuring. I also understand our immune system is quite different from mice, which is probably why so often why do we go to humanized models, whether they're organoids or organs on a chip or multi-organs on a chip? Some people try patients on a chip, and these chips get a little bigger. Why are these new microphysiological systems going hand in hand with this change or inflection point where we seem to be in working on disease biology and therefore on drug discovery? Since I mentioned the immune system, should we start with you?
Yeah,
we'll go in reverse order this time.
If you look at the amount of genes we have, right? We have exactly the same amount of genes than mice. But he looks different than a mouse. So why is that, right? But we have 270,000 splice variants. So we use these genes completely differently than the mice do, because they only have 40,000 splice variants. We have 270,000 splice variants. So what has evolved is actually not the amount of genes, but the utilization of the genes and the regulation of the genes. So that's why he looks different than a C57 Black 6 mouse. So that said, if you now say, look, let's do drug discovery things and disease discovery things in a mouse, you completely fail the mission, because if you do it with him and a real mouse, it's very different. So I think that's the wrong model system. It can be used to look at mechanisms. They may somehow overlap. There are good examples where you have a one-to-one correlation, but there's plenty of examples, especially in cancer, there's no correlation, you know. So I think we need to query diseases. We need to do discovery science in human diseases. The problem is we're not inbred, right? We're not all like the same. In the mouse house, they're all inbred. It means that you then take out one gene and say, okay, what's the effect of that gene in the background of the same genetics? But it also means these inbred mice, they miss a lot of other SNPs and genes that may be contributing to disease. So I think that's a stupid model to even do science on. We need it, but in the end, it's not. I think we cannot understand mechanisms of disease by just doing disease models in mice, because lots of things are missing.
Jumping around a little bit, Carol, you do a lot of organoid and maybe organs on a chip at Novartis, right?
Exactly, and there's a huge shift in human sample and data first. But this said, I'm going to come in here. We all agree all models are wrong. Some models are useful. And I think for what we need today, we need to characterize our model and know what to use for what, because the virtual cell is far away. And by the way, that's true also for AI model. They are wrong, but some are useful. Talking about organoid, one thing is to go human first, but the other one as well is to study perturbation in context. So if you perturb a cell line, you will see the intrinsic changes in the cell. If you perturb an organoid, every cell will be hit by that, but then the change in any cell will also affect the neighboring cell. And I think this humanized and in context is important. And finally, we're going human first because we can now, in some instance, generate human data based on reference data in position where we can't get access to it. And I think that's the whole promise of the field.
Anything you want to add, Steve or Fabian?
Yeah, I mean, let me say something in defense of the model organisms.
You as well.
Okay.
I mean, there's just certain aspects of shared biology that's not practical to do the experiments in human. Working out functional relationships often, because you can do perturbations in animals you can't do in humans. It provided enormous insight and discovery over the years. And it's a question of, I think, being selective about the questions you're asking, and asking questions about the universal biology that's shared across organisms. When we at CZI were building the first large language model, or a large language model transcriptomer on the Cell Atlas data, there was a big internal debate about whether it should be human only or across model organisms as well. And I pretty much put my foot down and said, we're going to do this across the evolutionary tree of life. And people didn't understand why at the time. But as we got into it, what we found was that we could use it to try to predict how experiments would transfer from model organism to human. Okay, so when you train across the whole spectrum, you can use it to try to predict what will translate and what won't from model organism to human. And I think these would be a great example of how AI can help us use model organisms more intelligently. I will say another example I've been involved in. I mean, in my lab, we've developed a number of liquid biopsy diagnostics, all that human from the beginning. So in that case, we wanted to be connected to biology. It wasn't difficult to do the experiments. It was all about getting the human data. So none of those were done in any animal models. In therapeutic development, the case I've been involved in, both matter. I mean, there have been times when you just can't extrapolate, and it's going to be quicker to get the data in human, particularly around tox and things like that. There's only so much you can infer. Whereas for other things, like for cancer therapeutics, what combination of therapies might work, but too expensive to do in human first, and so you want to do some xenograft mouse studies to get an idea of which direction to go with the human trials.
Yeah.
Maybe I can briefly extend sort of on the method part here, because I think, let's say we want to build a representation of just genomic variation, right? Like a language model of DNA, if you want, right? I think it's very clear that even though we have a lot of variation, right? I mean, what's our similarity? 97, 98%, or even more, like super high, right?
We haven't checked yet.
Well, it was good that I dressed up this morning, that you noticed that I look different from Black Six mice. So this variation, in order to model like just a big DNA model, you need to do tree of life, exactly that. People like, there's a colleague at our place, he just goes through all fungi and sort of tries to understand how sort of various transposable organisms, whatever sort of comes together in order to really just build that basic core grammar of DNA. So I think that's very clear. Having said that, I think what we want to do is not just sort of the description of the state on our map, but also sort of how we move, sort of this effect of perturbation, this sort of where we go. This is going to get super large anyway, so I think it won't work without transfer learning. What we've been doing since the beginning of human experimentation is, and that's why I'm always fascinated when I talk to my experimental partners, like you have in your brain like this network of things, this is the next experiment that I need to do. So like any scientific theory is something that predicts how a model behaves under a perturbation, right? So that's why, if you know in a model system that there's maybe something, maybe you sort of draw this sort of maybe even serendipity-based type of parallel to say what you want to do in the human. So I think having some of these, we can even do it serendipity, we can even do the sort of computational to put some of this data together. But I think eventually this, like measuring what these things are, the validation, we need to then ideally do in something simpler. It could be organoids, it could be something in between.
Yeah, and maybe I can just add to that, coming back to one of your points, yes, we are far away from the human virtual cell to inform the clinic, yet AI is usually very good at one thing, is predicting what doesn't work. Now, if you think about a clinical team who are looking at what dose to give to patients, these models, even though they are not perfect nowadays, they can still tell them what not to do and what a okay starting point is. Obviously, the more you progress in the clinic, the more accurate you have to be. But for the first in human, they're actually already really useful.
Maybe just two more comments, rebounding on some of the things that have been said. On the animal models, I thought, well, maybe we don't fully understand the efficacy or the mechanism of action between mice and men. But then it's even thought, well, for toxicity, they must be pretty good, right? And it's absolutely remarkable. It's something as basic as DILI, as drug-induced liver injury. Sorry, that's what DILI stands for. How much of that is tested in multiple mice and in one more animal model, and then there's immediate human interaction or liver toxicity. I thought something as basic as that you would get from the mouse model, but very often it turns out you don't, so there's some limitations. And my other comment, you mentioned splice variants. Well, I'm a proteomics guy, so post-translational modifications, isoforms, glycosylation, the complexity when you get to the protein level, the proteoform level is enormous, the heterogeneity, and not only between humans, but also us longitudinally, our different cells, if we have cancer cells or rogue immune cells, I assume, their glycosylation and other post-translational modifications are enormously different. That's where some of the limitations are. We don't have good lampposts that illuminate that and elucidate for us today. So there are many things that we can still do better, but at least, as you said earlier, Carolyn, we have a better understanding of what we don't understand yet. We maybe approach biology and disease with more humility today than 25 years ago, which is a good thing. And we have a lot more information. And we have this big 800-pound gorilla called AI in our corner who can really help us, give us a chance with all of that, with the deep learning. So good perspectives, thank you. At this point, we have a good 15 minutes for your questions. If you will bring a microphone to you, please briefly say who you are. Make sure you give us a question, not just a statement. And I'm old-fashioned, I'll start. Okay, we start with the gentleman over here, and then we'll go to the ladies. Yes, sir, please.
Hi, I'm George Caputa. I'm senior editor at Nature. And I have dozens of questions.
I'm sure you would.
But I think that... no, it's Friday. I think that something, the preclinical and basic biology space has just exploded with AI and access to data. And I think we do a lot of talking about, as a clinician, standing in a hospital, what are your options for treating someone that's standing in front of you? We really need clear, I think, if we go back to how do clinicians get clear direction as to what they can use, and those are from clinical trials. phase 1, phase 2, phase 3? Is it safe? Is it effective? Is it effective in a broad category of individuals? Do you think that the way that we are testing things through clinical trials and the design of clinical trials in terms of the numbers of people and how people are selected, do you think that really reflects the reality of the data that we have now? Because, you know, something that's so powerful is you have, you know, at Nature, we see these drugs come
Maybe let me get to.
Yeah,
Can we
but I'm
take
just
that
saying, like, you
question,
know,
run with
like
that question?
60 years of research, and then all banking on this one moment.
Yeah.
You know? So I'm very interested to hear what you all think about how you can use the data that you're
You're
kind of
chomping at the
inform
bit,
that.
Carolyn.
Just to your question in particular on whether we select the right patient for the right option, my answer is right now not optimally, because we select the patient based on document and patient history, and we try to match it to molecular understanding. And I think that's about bringing together the phenotype of the disease with the mechanistic understanding.
I'll give you an example. If you have a little bit of fever and then your knee is swollen and the doctor says, oh, this is RA, what do? Give you some drugs, but then give you eventually TNF inhibitors because it works in 50%. And they say, okay, in 3 months come back and we'll see whether it worked. So that's empirical, right? So most of the compounds and drugs that are given are empirical. Actually, TNF, when first tested in sepsis, was a nightmare. You killed people. And this was almost the end of biologics, right? And there was a rheumatologist taking the drug and giving it to a patient that was really sick, and he got up the next day and was fine. So this opened up the entire field. It's all empiric. That's the problem. It's not all, but I think what should be done is you have symptoms and give the patient, and you control your own symptoms towards the treatment response. That's what you do in genetic diseases. But we are treating Gaussian distribution curves of patients that are phenotypically selected and not mechanistically selected. That's a big issue.
Yeah.
I'll go to the next question, and then I'm sure. Go ahead, please. Yes.
Thank you very much.
Would you get up and introduce
Yes.
yourself?
My name is Katrin Tedik. I work in Essen, Germany at the research center One Health, and I'm very happy that I get to ask my question about after the Nature senior editor, because I wanted to follow up on your commentaries on the models that we are using, because I agree that we should measure more in humans, and there are extremely powerful public data out there, for instance, in TCGA, right, and also other cohorts. And my experience is that when we use human data and we use them to validate mechanisms that we find in model systems, and we put them in our manuscripts, the reflex from the editors, including Nature,
and
is
the reviewers.
to ask me, no, actually not the reviewers so much,
Okay.
it's more the editors, who then say, oh, it's really great you show this in humans, but can you also show it in a mouse? And yes, and I mean, this of course reflects also on science policies in the US versus Europe, where it's becoming more restrictive, doing mouse work. But I think it's really important that this changes and that we can measure more in human cohorts, and that using public data is gaining more values over the mouse models. And I don't
Did you want to
wanna
finish with a
discredit them.
question, or?
Yes. So what's your take on this? So what do you think, where we stand with the human cohorts and with the public data? How much can we do in there already, and where do you think are the data gaps?
So
Fabian, and
I
then
can briefly
Steve,
take
maybe?
the data question. So I think basically also what George said, right? We're going to have, at the moment, very often we have one data set, one project, one paper, right? And we sort of typically move on. And I have the impression sometimes in pharma is like, if you have an old trial, you might not have measured this one or the other modality. You don't sort of put it into sort of one bigger thing. But we've learned, right, in these sort of big foundation frontier type of models that we can kind of eat up the whole internet in terms of text and sort of just sort of suck it, smooch it together and, do actually something meaningful with it, right? And I think this is happening more in this. So I think clinical trial design, beautiful point, right? I think this is majorly changing. A lot of actually cellular AI people, that there's a few big shots that moved to pharma. They actually didn't stay on the cellular side, but they actually used that information for clinical trial design. A whole bunch of startups go in that direction, where you can reuse then some of these sort of more basic things, some of their own trial, and then design new ones. So I think we're going to see a lot of this coming out.
Steve, I'll send things over to you, but maybe one drive-by comment. I think always asking for animal data when you find something in humans is nonsensical, because their biology, including their disease biology, including their immune system, sometimes will be so different that you may or may not find it in animals. Steve, over to you.
Yeah, I agree with that. I don't have a lot to add except to say that we're entering sort of more powerful era of using human cohorts because of shared medical records and things like that. So the ability to do discovery in humans is at an amazing time. And so for many things that previously required animals, we can probably go right to humans.
One more comment,
and then the lady.
So
the microphone, hopefully.
Big pharma has come together to write papers on particular mouse models that did not translate, because even if 5 different pharma companies take one mouse model with one gene identified as a target, it doesn't translate in the different places. So this is a little bit of a problem, because the microbiome has such an influence on disease phenotypes. So I think we have to really reflect on that. So we are using we're using Black 6 that are 2 years old, and we're just taking 500 mice as a discovery approach and look only at the variants. The variability of the phenotype is drastic. There's mice that are completely inflamed, has tumors, others are completely healthy, they have no gray hair, so some mice have gray hair, some are bold, some are not. And you can find out actually what makes boldness by just looking at the diversity, which is not genetic in this case, it's the microbiome changes. So that's an in vivo discovery approach that really makes sense.
We have 2 more questions. I think you already have the microphone, then we'll go to you.
Yeah. Hello, my name is Maria Chernachenko. I'm probably one of the younger generations here. I'm doing my PhD in University of Toronto. And as a part of my program, I have to teach, and I'm teaching a stem cell therapies course. And a question that my students often ask me is, why don't patients receive stem cell treatments in earlier stages of the disease? Because what we often see with stem cell therapies is that they're only allowed to be tested in patients that are terminal, where the potential benefits of the treatment will overcome the risks of it, and there is no other treatment available. But often in neurodegenerative disease, in a lot of degenerative disease where stem cells could be really useful in early stage, they're not allowed to be used.
Why not stem cells earlier?
Maybe I'll start, and I'm sure you got something to say.
Yeah, just my point of my question is, how can we make it a safer and more approachable method to test certain therapies early on?
Steve, you can take that. And
so I think probably the most powerful aspect of stem cell therapies has been bone marrow replacement, right? Where if you've got cancer of the blood, you can get a new immune system. And that's an amazing success. I think it's been harder to show efficacy with other stem cell treatments. I mean, there's a lot of snake oil out there where people are doing injections in your knees for issues around there, where it's been very hard to show efficacy. And so I think it's just without the evidence, it's not going to pass medical standards.
Yeah, it's exactly that, but also cost and commodities. It's very hard to make always the same stem cells. And
does it not work, or is it toxicities?
It just doesn't work. And there's probably toxicity too, but it's not working. I mean, you'll tolerate more toxicity if you're getting positive benefit, but I don't think it's been hard to show benefit from any of those.
Are there immune problems if you get stem cells from other people and stuff like that?
Absolutely, but you brought BMT into play, so you can use bone marrow transplantation in rheumatoid arthritis, in all these immune diseases that are autoimmune, like antibody mediated. It works, but it also kills a few people. So it's a risk-benefit problem here, right? Now we can actually selectively deplete B cells, and it's actually a revolution in medicine. You can cure lupus, for example, like a deadly disease. You can cure now with a single injection of CAR T cells. But even that is not a commodity. It's so hard to take cells and make this in vitro, and we ship them around in the world to get the right cells. But it's being done at the moment, and this is it's a field that is evolving. But now we can do this in vivo, inject something, so you make your CAR T cells in vivo, or take bispecific antibodies where your T cell kills the B cells. All these things are happening, but it's not a commodity yet.
I did observe that I heard anecdotally from a friend recently who is into this longevity research where you can get certain stem cell injections from, I'm told it's from Mormon women that don't smoke and drink, so that's good. And you can get it in Tijuana, just south of the border, where the FDA and maybe these things work. People come back rejuvenated. I don't know how much of that is psychological or not. But next to your question, please. Yeah.
Thank you. Hi, I'm Verena Haig. I'm a neuroimmunologist, and I work at Charité right now. So my question is related to how far are we really with this multimodal and multi-omics data integration? Because we have all these single-cell RNA-seq data sets, cell-seq data sets, proteomics data sets, omics, epigenome, and so on. Exposome is a big thing as well coming up. And I'm part of a taxonomy group trying to actually settle down on a number and how to define microglial subsets right now, which already takes 2 years and is also funded by the CCI. But so I'm just wondering, how far are we really if we as a microglia field take 2 years to identify how many subsets we have and we want to understand global disease and by integrating these
lots of silos, lots of walls that need to come down, but this gentleman is working on bringing them down. Aren't you doing this?
No, he's doing it.
Multimodal. Okay. Anyway,
we just make the samples. He analyzes them.
You're measuring them. I mean, I might start, and maybe Steve, you can chip in because the Tableau, of course, examples are fantastic. So when we started, and I just sort of started maybe with the RNA side of things, right? When we started building these first atlases, there was basically, I think there were 30 papers around that said we have built a lung atlas. And it was always just N equals 3 to 5 patients or subjects, right? And then you sort of sample a little bit different pieces, and these things sort of just look completely different. So the typical thing, whenever you look at high dimension data, what you always observe is that the major sort of source of variation is going to be where you sample, lab variation, and so on. So you need to have tools to bring this together. But I think the beauty of sort of advances in machine learning are that nowadays we know how to bring things together. And this now I'm just talking about one modality, but if you know how to sort of fuse this, of course, let's say if you have here 2 modalities, here just one, you can use this as a bridge and then sort of transfer over that additional modality to that. So when we build our first integrated human lung cell atlas, right? So we brought, I think, together up to 50 data sets now. We make a V2 where we have all kinds of diseases on top, where we really have then this observation. That sort of opens up really interesting new things where, in 2 diseases that you don't sort of look into each other, like maybe just randomly, maybe not. And I think we saw something like in monocyte-derived macrophages, there was one factor high up that was associated both in lung cancer as well as in COVID, because it was some type of scarring thing, which maybe even opened up new ideas for targets. So I think there's sort of these serendipity-based new things that you find there, which is kind of cool. And now you ask, like, when we, on the other hand, take so long with annotation, sort of curation, I think this is what you said with your sort of cohorts, with your sort of internal analysis, and be similar for us, annotating those clusters then takes really long time. Just this one point I want to make is that biology obviously doesn't care about what we call those things, right? Do we have now, I mean, when we started, we said, do we have 10 cell types in the brain or 1,500 as Stan Linderson or Alan Brain now claims? I mean, it's just like groups to different scale. We need to give them names, I guess, because otherwise every immunologist has their own sort of favorite name.
Exactly.
CD4, whatever. But I think the biology and maybe also the machine learning systems don't care so much about it. So for us to look into it and understand, sure, right? But I think for doing some type of prediction, it might not even matter that much. So you can even go ahead without maybe sort of fully describing things and giving nice names to things.
Any other comments, or should we take one more question?
I could maybe add,
If you could give the microphone back, please.
So to the question on omics, which is the best omics? Sidney Brenner was asked that once. What's the best omics? Proteomics, genomics, metabolomics? He says economics.
Oh, nice.
But to your particular question of cell type annotation, there's ways to do that from first principles. And in fact, Urilan and I have a preprint up on using Pareto optimality to do that, and I'd encourage you to check that out, because humans they're sort of motivated to find many different subtypes that may not be there. Immunology is notorious for this, because every new cell type was a new paper. So there was kind of a perverse incentive.
Okay, almost out of time. Fastest question ever.
Ashokesa from K. Leuven. I'm in the Skeletal Biology Engineering Research Center. I'm speaking fast. I'm working in osteoarthritis model and non-union models, and we are trying to take it to bedside. So I'm going to ask you on the perceptions on the ATMP productions on the manufacturing side, because we talk about bench. Now I wonder your ideas how to take it to patients in the ATMP perspective.
Well, in our last minute, who can answer this one?
No one.
No one.
No.
We're all looking at you as the farmer rep.
See, we're still in the dark ages. Well, that was a downer at the end of all of this.
Discussion. Longer discussion. I don't even know where to start to answer your question. So maybe we take that
Offline.
offline.
The answer is we do not have all the answers. Thank you very much. I think this was most inspiring and a vibrant discussion. I'd like to thank the panelists very much for their contributions. We thank you for your attendance, and let's bring down some more barriers and walls. Thank you very much.
Thanks, Mark.